Life is movement, and movement requires energy. From the chemical reactions in your cells to the beating of your heart, every process in biology is powered by the transfer and transformation of energy. We begin with the basics: kinetic versus potential energy. Kinetic energy is the energy of motion, a rolling ball, a flowing river, or electrons moving through your cells. Potential energy, on the other hand, is stored energy, like water held behind a dam, or chemical bonds waiting to be broken. Life depends on the constant conversion between these two forms. This brings us to the laws of thermodynamics. The first law, the law of energy conservation, tells us that energy cannot be created or destroyed, only transformed from one form to another. The second law, the law of increasing disorder, states that every energy transformation increases entropy or disorder in the universe. That's why no process is 100% efficient. Some energy is always lost as heat. In biology, chemical reactions can be grouped by how they handle energy. Exergonic reactions release energy, like cellular respiration breaking down glucose. Endergonic reactions require an input of energy, like photosynthesis building sugars. Cells constantly couple these reactions so that energy released from one drives the other. But reactions don't just happen instantly. They require an initial push called activation energy, the barrier that must be overcome for bonds to break. This is where catalysts like enzymes step in. Catalysts lower the activation energy, making reactions proceed faster without being consumed themselves. Enzymes are remarkable because they are highly specific. Their active site is the precise region where substrates bind and the reaction occurs. Sometimes you'll hear the term substrate binding site. This refers specifically to where the substrate attaches, while the active site encompasses both the binding and the catalytic activity. Enzymes are also sensitive. Their activity depends on temperature and pH. Too much heat can denature an enzyme, unraveling its structure, while extreme pH levels can disrupt bonds and interfere with function. Each enzyme has an optimal range where it works best, and even small changes outside that range can slow or stop its activity. Cells also regulate enzymes. Repressors can bind to allosteric sites, regions distinct from the active site. This changes the enzyme's shape, preventing the substrate from fitting and effectively turning the enzyme off. It's a form of molecular control that ensures reactions happen only when needed. Finally, no discussion of energy and life is complete without ATP, adenosine triphosphate. ATP stores potential energy in its phosphate bonds. Each phosphate group is negatively charged, and holding them together is like compressing a spring. When a phosphate bond is broken, that stored energy is released and used to power endergonic reactions throughout the cell, muscle contractions, active transport, or synthesis of macromolecules. ATP is the universal energy currency of life, constantly being spent and regenerated. From energy's basic laws to the elegance of enzymes and the power of ATP, life is a story of capturing, converting, and controlling energy. Every heartbeat, every breath, every thought is possible because of these principles. And as we'll see, the flow of energy is what connects every living system on Earth.